This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only.
We all strive to live a long, prosperous, and healthy life. With advances in health and medical sciences, this goal is ever more attainable. The Sam and Rose Stein Institute for Research on Aging is a non-profit organized research unit under the auspices of the University of California at San Diego, committed to advancing lifelong health and independence through research, education, and patient care. To better empower and improve the lives of young and old alike, the Stein Institute presents the following program.
Without further ado now, I'd like to introduce tonight's speaker. As I mentioned, we're really fortunate to have with us tonight Dr. Paul Eisen, who is a professor of neurology and medicine here at UCSD. He is the vice chair of the Department of Neurology and the director of the Memory Disorders Program at UCSD. When Dr. Eisen came to UCSD in 2007, he came as the director of the Alzheimer's Disease Cooperative Study, which is a consortium that's funded by the National Institute on Aging to develop methods and conduct clinical trials in Alzheimer's disease. He is the highest funded neuroscience investigator in the country, as is our Department of Neuroscience here at UCSD. So as I mentioned, we have the man here with us tonight. So his own personal work relates a lot to investigating therapeutic approaches to treating Alzheimer's disease, from secretase modulators, amyloid binding compounds, cholinesterase inhibitors, and nicotine antioxidants and anti-inflammatory drugs. He is an extremely well-credentialed gentleman. Before he came to UCSD, he was the director of the Memory Disorders Program at Georgetown. He received his undergraduate degree from Harvard, his medical degree from Columbia. He did residency training at the University Hospitals of Cleveland, as well as New York University and Mount Sinai. He is a diplomat of the American Board of Internal Medicine with specialty certification in rheumatology and geriatric medicine. So without further ado, and without going through the other numerous credentials that he brings to the table, I'd like to present Dr. Paul Eisen, who's going to speak with us about the aging brain.
Okay, well, thanks for joining us this evening. I'm going to talk in general about the aging brain. I'm going to start by putting it into context of aging in general. I'll talk about how aging influences the brain and brain function. So I'll talk about aging, the aging brain, and the most important disease of the aging brain, which is Alzheimer's disease, tell you a little bit about what we're doing here at UCSD, and in general where we are in trying to better treat Alzheimer's disease. So I think you know that aging is an increasing problem, which essentially is good news. Longevity is increasing, but the number of individuals at an old age in this country and around the world is growing rapidly, and that has major implications for everything, as you know, but in particular for age-related disease. And the most important, to my thinking, age-related disease is Alzheimer's disease. And since our population is getting older and the major risk factor for AD is aging, the number of cases is skyrocketing. So we have about 5.5 million cases today, and just on the basis of the population getting older, we project 14 million cases in the US by the year 2040. So as big a problem as it is now, it's going to triple in the coming decades. Here you just see shown graphically that portion of people who are over 65 and over 85 is just going steadily up. So all conditions related to aging become much, much more important. Why is our population aging? Of course, it's because life expectancy is rising. Life expectancy in the mid-19th century was about 40 years of age. At the beginning of this century, it was 78 years of age, so a doubling of life expectancy. Now, why has that happened? mainly because of public health improvements, nutritional improvements, some medical advances like antibiotics and vaccinations, and some lifestyle changes. For example, smoking cessation is probably a big contributant to increasing longevity these days. Better medical care probably plays some role. Looking at the graphs I just showed you, you might ask, well, how much longer can we live? Are there limitations? And there are, and I will talk a little bit about that. But of course, in this Stein program, the focus is successful aging, and I want to emphasize that the goal is not just quantity of years, but quality of years. Now, why does our body suffer so much as it gets older? From an evolutionary perspective, it's easy to understand. Evolutionarily speaking, the body was designed to reproduce. So past reproductive years, there was not much evolutionary pressure to do better physiologically. But we can understand the deleterious effects of aging, what happens when evolutionary pressure is no longer improving the species because individuals are beyond reproductive age. We can understand this by looking at the biology of the impact of aging on the molecules in our bodies and the cells and the organs. And I'm going to tell you a little bit about that. Longevity is related to genetics, and I won't talk too much about that, but I will point out that life expectancy is somewhat hereditary. We see this in studies of identical versus fraternal twins, and we see it in that having parents who live to an old age is a predictor of an individual living to an old age. Women, as you know, live longer than men. So genetics and gender contribute to longevity. What happens? What has the biology revealed to us about aging? Well, here, just as a brief introduction, I point out some of the biological aspects of aging. And we usually, in geriatric medicine and gerontology, we study aging at all of these levels. So the level of molecules, the major building blocks of life, DNA and proteins and all of the large cells of life become damaged related to age. So if you study molecules in individuals who are older, you see that their structure changes and their function changes, and that contributes to aging of the organism. So at the level of molecules, we have learned a lot about the chemical aspects of aging. At the level of cells, we also see that cell death occurs on a genetic and toxic basis, and we can study the aging of an individual by looking at changes in the cells of the individual as the person gets older. And probably you're most familiar with aging at the level not of cells but of organs in the body. And here we see, for example, that the musculoskeletal system weakens, and I'll talk a little bit more about that in a second. The heart thickens, its ability to respond to the need for more blood flow is impaired as you get older. So the changes to the molecules and the cells contribute to a loss of resilience in the heart and the lungs and the kidneys that contribute to the aging of the individual. So molecules, cells, organ systems all show a increasingly well understood decline in function with age. It contributes to the longevity of the organism, so the lifespan is limited by these changes at all these levels, and at the level of species, this is the longevity of humans, currently around 78 to 80 degrees. We've learned a lot about all of these processes, but yet can't put the whole story together yet. So we still don't understand which of these contributes greatest to the overall aging of the individual. Now, I'm going to delve into the science a little bit before I step back out again. In talking about why a cell and an organism get older, age, and is there something we can do about it, I want to suggest to you that there's a limit to what we can do, and we understand this now at a molecular and a cellular level. The same processes that the cell needs to survive limit the cell's longevity, and by the same token, there is likely to be no way of continuing to increase longevity of an individual. So the example of this that I've put up here is a particular gene that creates a protein called p53. So this is a molecule in our cells, and we now know that this p53 gene helps protect cells against cancer. That is, what cancer is at the level of a cell is uncontrolled growth, where the cell keeps dividing so that a tumor is formed. The same gene that protects against cancer, protects against uncontrolled cell death, uncontrolled cell growth, formation of tumors, plays a role in aging. If you eliminate this cell, if you eliminate the gene p53, this tumor suppressor gene, the cell becomes cancerous. But if you increase p53, you can block cancer, but then the cell ages too quickly. The two processes are inversely related, and I show that a bit on the next slide. So we can study this one gene, this tumor suppressor gene p53, we can study it in cells, and we see that cells express it at a level that balances risk of cancer and risk of aging. And if we increase the amount of this gene product, we can protect against cancer but increase senescence or cellular aging. And by the same token, if we adjust this gene product to prevent aging, then the cell becomes neoplastic. It turns into cancer. So there is a balance between controlled growth and cancer that relates to aging. And we can't have too much protection against aging without an increased risk of cancer. Now, is there anything that we can do to increase the likelihood of living to 120 instead of living to 70? I don't know if any of you have any ideas on what the current thinking is regarding the best strategy to promote long life. Well, not much you can do about your genetic background, so what can you do something about that could... Yeah, exercise is a good answer. What science suggests is the best answer is eating less, caloric restriction. So in animals, restricting caloric intake to keep the animal skinny is the best way of increasing life expectancy. Whether this translates to humans is unknown, but there's a very interesting science growing up around the idea of caloric restriction, and the impact of caloric restriction on mice is very clear, promotes long life. On humans, not so clear yet, but this is probably the leading candidate intervention to increase lifespan. Why does caloric restriction seem to be good for animals? Well, it seems to slow down metabolic rate and therefore slow down the accumulation of toxic byproducts of cellular metabolism. So essentially you're slowing down the function of cells and organs in the organism, and that allows the organism to live longer. But while this is proven in animals, it's not yet proven in humans. I am not suggesting that you eat less. Now, I'll say a little bit more about what happens to your bodies as you get older, or what happens to your anatomy. I don't think this will be a big surprise. There are striking changes to your anatomy as you get older. I've just highlighted here the loss of height. your height peaks between ages 30 and 40 and then declines. How can that be? How do you get shorter as you get older? It's because of changes in your joint structure and function, so that your hips become bent and your knees become bent. So it's not that your bones are getting shorter, it's that your posture changes, and the result is a reduced height. Now, what happens to your muscles? Well, your muscles atrophy, they become smaller, they become less powerful, your bones become thinner. So aging has as profound an effect on the musculoskeletal system as on any organ system in the body. What can you do about aging of the musculoskeletal system? Here is where exercise is clearly the answer. Staying active, walking, exercising is the best way to improve muscle mass and muscle function and bone density, reduce the risk of fracture, and maintain function. And I mention this not because this is a talk on the musculoskeletal system, but we may guess that the best way to protect brain function is also to increase brain activity, to maintain the function of our brain cells, maintain the function of our brain connections. And yes, exercise is under intense study now as an intervention against brain aging. Now, before we move into the brain, I just want to make sure I make the point that as our society is getting older, the effort to market remedies to our society has grown without bounds. And so if you go into a GNC or a CVS, you see whole aisles devoted to anti-aging remedies, nutraceuticals, immune system boosters, brain boosters. And I want to make sure that I tell you that there is no evidence for any of these, that there is nothing you can take by mouth that slows the aging process or improves your brain health or boosts your immune system. So this is just marketing. Now, the Stein Center is focused on successful aging. Successful aging is the process by which we minimize the adverse effects of aging to optimize physical and mental function. Living long is not sufficient. We want to live long with the best possible quality and the highest possible function. How do we accomplish this? Really don't know. So it's not going to be found in a GNC store. The extent to which we can promote successful aging with exercising our bodies, exercising our minds, maintaining a vigorous, interesting, creative lifestyle, we hope that this helps. It's plausible that this will help, but specific interventions really remain to be evaluated. So let's turn to the brain. That was sort of the general context of the aging body. Let's talk about the brain. And here I want to tell you a little bit about the normal aging of the brain and then Alzheimer's disease. So aging of the brain is inevitable, and there are changes that occur in everyone. But Alzheimer's disease is not inevitable. It is a very common disease, but it is not an aspect of normal aging. It is not inevitable. What's the effect of normal aging on the brain? Well, first, just like your muscles, your brain atrophies. It's actually a fairly notable degree of loss of substance that occurs as you get older. The brain of a 70-year-old has lost 10% of its mass compared to the brain of a 30-year-old. So the degree of shrinkage of the brain is quite significant. And this reflects a loss of brain cells and a loss of functioning brain cells. The chemicals that allow communication between brain cells decline, and we understand a bit of why all this happens. It has to do with the change to molecules and change to cells during the aging process. But the net result is a shrinking brain and a loss of brain function. What do I mean by a loss of brain function? I think just about everybody in this room is aware of this, because typically by age 40, you're pretty sensitive to the changes in your brain function. Normal aging of the brain causes slowing of reaction time, constriction of working memory, difficulty in multitasking. There are changes that can be tracked. Your brain function overall peaks around age 20, and it's downhill from there. And by the age of 40 or 50, people are very upset that they can't remember the right word or the right name anymore. But this is not Alzheimer's disease. This is inevitable, normal aging of the brain, manifestations of that general atrophy, that general shrinking of the brain with age. Brain function declines, but there are certain important aspects of brain function that are preserved despite the aging process. For example, communication skills are not affected. Older people, and I'm relieved about this, older people are still able to communicate effectively, and in fact, vocabulary tends to continue to improve. Even if pulling the right word out is always a problem as you get older, overall understanding of language, comprehension, tends to improve as you get older. The effects that I just mentioned, the slowed reaction time and decline in working memory, these are annoying, but the good news is that in the absence of disease, these changes really don't mean a substantial loss of function. So they're annoying, you notice them, you worry about them, and you can rudely fail to mention someone's name when you see someone familiar, but you're able to compensate in terms of function, live independently, enjoy reading and music, communicate effectively, and in fact, comprehension tends to improve. So overall, the good news is that despite the aging of the brain and the change in function that that aging entails, brain aging does not have a substantial deleterious effect on function in the absence of disease. But disease of the aging brain is highly prevalent, and the most important disease of the aging brain is Alzheimer's disease. And what I've just told you about normal cognitive aging is certainly not true of Alzheimer's disease. The cognitive disturbances of Alzheimer's disease have a profound effect on function, and they are steadily progressive. This is a serious, highly prevalent disease of aging. How prevalent? Alzheimer's disease affects about 1% of people in their 60s, about 10% of people in their 70s, and about 50% of people over the age of 85 to 90. So highly, highly prevalent. And that's why, again, as our population ages, as the proportion of our population over the age of 65 and 85 keeps increasing, the impact of this disease keeps increasing. Now, one good thing about AD is that we understand it a lot better now than we did a few decades ago, and we now have treatments, but we have a long way to go. And what I want to spend the rest of my lecture time with you on is where we are in Alzheimer's disease therapies. That's what my work focuses on, and I want to kind of review the situation for you. So what is Alzheimer's disease? Again, first it's a disease. It is not what we used to think, which is that it was just getting old, senility. It is not senility. It is a specific disease with specific causes and specific changes in the brain. We call it a neurodegenerative disease. It's not caused by an infection. It's not a metabolic abnormality. It's a disease of aging. What's the relationship between Alzheimer's disease and dementia? Well, it's the difference between a specific disease and a syndrome. So in medicine, a syndrome is a collection of symptoms that occur together, typically from one of a number of possible causes. So for example, arthritis is a syndrome that can be caused by gout or rheumatoid arthritis or other forms of arthritis. Dementia is the syndrome of cognitive impairment having a negative impact on day-to-day functioning, and the most common cause of the dementia syndrome is Alzheimer's disease. So Alzheimer's disease is a specific common cause of dementia. Now, everybody over the age of 40 or 50 is aware of their aging brain and the changes that accompany the aging of the brain. How do you distinguish early signs of Alzheimer's disease from normal aging of the brain? Well, the most specific indication of Alzheimer's disease as opposed to normal aging is a deterioration of the ability to learn new information. So that's different from being able to pull out a word or a name. It means that if you have a conversation, an hour later you're going to have trouble recalling that conversation. The information contained in that conversation was not encoded in your brain. That's the most specific difference between Alzheimer's disease and normal aging, is the marked impairment of the ability to learn new things. And this manifests itself not only as the difficulty in recalling recent events, but a tendency to just ask the same questions over and over again because the answers don't get recorded in your brain. So this impairment of learning even more than memory, is what separates Alzheimer's disease from normal cognitive aging. Now that's Alzheimer on the left there. In the upper right corner is August D. In Alzheimer's time, which was the early 20th century, scientists used to give pictures of their patients and even partial names of their patients. We don't do that anymore. So August D is famous as the first person identified with this disease by Alzheimer. And she was a woman of 50 years who had a progressive cognitive and behavioral disturbance. Alzheimer was a psychiatrist and brain pathologist. He treated her and then examined her brain, and we didn't have the ability to photograph through microscopes, but he did look through microscopes, and he drew pictures of her cells. And that's what's shown in the lower right there. And what Alzheimer noted is that August D in her brain cells had these dense fibers dividing the cell bodies. And those are called neurofibrillary tangles. They're dense rope-like accumulated material within the cell bodies. Alzheimer drew them in his first paper in 1906. And in fact, his drawings are a pretty good representation of what we see today with our modern microscopes. This is the overall picture of the brain with AD. Now, I said that a normal brain, as you get older, loses about 10% of its weight from young adulthood to older age. In the AD brain, that atrophy is accelerated, but it's also regionally specific. So it's not the whole brain atrophying at the same rate. If this is a cartoon of the normal brain, the atrophy in Alzheimer's disease is particularly prominent right here, as you see those big cavities growing where they were not present to any significant extent in the normal brain. These are the medial temporal lobes. That's the learning center of the brain. So the reason you have this impairment in learning is that the atrophy, the damage in the Alzheimer's brain, is specifically prominent in these medial temporal areas that are responsible for learning. Now, here's a photomicrograph, what Alzheimer didn't have, but we have now, showing a slice, a microscopic view of a slice of Alzheimer's disease brain compared to normal brain. And here you see all these dark areas. Those are the nuclei that indicate cells. And the number of cells here is markedly reduced. And in addition, the cells have dense changes, which are the tangles that Alzheimer drew. And in addition, there are these reddish deposits that are called amyloid plaques. And Alzheimer described these as miliary changes in the brain, meaning widespread small deposits. We now call them amyloid plaques. At higher power, this is what those plaques look like. They're deposits of a very dense fibrillar material in the substance of the brain. So the brain becomes dotted with these deposits in Alzheimer's disease. These are the amyloid plaques, and the neurofibrillary tangles that Alzheimer drew are these dense bands within cell bodies, and here we see them with different staining methods. So the two characteristic changes in the brain in Alzheimer's disease are the amyloid plaques outside the cells and these neurofibrillary tangles within the cell bodies. Now, sketching very quickly the history of our understanding of Alzheimer's disease, there are some interesting lessons here. I told you that Alzheimer wrote his first paper in 1906. Now remember that I told you at the beginning that life expectancy in the mid-19th century was around 40 years of age, not so different in 1906. So Auguste was actually an older person for that part of our history at the age of 50, but it led us astray. So there weren't very many people in their 70s and 80s then, and the first case described happened to be a woman in her 50s. And for the next 3/4 of a century, we called Alzheimer's disease pre-senile dementia. It was thought to be a disease not of old age, but of middle age, based on that first case. So the general feeling is that everybody has a tendency to senility, just as an aspect of aging. But if you become cognitively impaired in middle age, well, that's Alzheimer's disease. And it wasn't until 1976 that a neurologist named Robert Katzman, who became the first chairman of neurosciences here at UCSD, convinced the world that there's no such thing as senility, that senile dementia is in fact Alzheimer's disease, that it is one disease with the same plaques and tangles whether you're middle-aged or older, and in fact, it's not a disease of middle age, it's a disease that can start in middle age, but the prevalence skyrockets as you get older. So it is not inevitable to have senile dementia. It is simply that Alzheimer's disease becomes more prevalent as you get older, but it is a specific illness. It is not just a consequence of normal aging. So it was Robert Katzman, as recently as 1976, who convinced the world that Alzheimer's disease is not a rare disorder of middle age. It is one of the most important healthcare problems in the world, a highly prevalent disease of aging. That demonstration by Katzman markedly increased the level of interest and scientific study in this disease, as it was recognized that this is a highly prevalent, serious world health problem. That led to a huge number of scientists trying to study the chemical basis of memory loss and the discovery of a specific chemical that mediates memory, and that's acetylcholine, that ultimately led to the treatments that we have today. So Alzheimer's disease is a disease characterized by these two changes in the brain, the amyloid plaques and the neurofibrillary tangles, but the net result of those changes is damage to cell function. And cell function that's necessary for memory is actually a chemical communication between cells that involves the specific chemical acetylcholine. And we can today use medications to increase the amount of acetylcholine in the brain and partially compensate for the memory and learning impairment of Alzheimer's disease. So recognizing how prevalent the disease was led to a huge amount of work that changed our view. Our view up until then had been that memory is not the kind of thing that can be treated. But that turned out to be wrong and led to the drugs that we have today. In the mid-'80s, while these treatments were first being explored, George Glenner, a UCSD scientist, UCSD has been kind of at the center of Alzheimer's disease for the last 30, 40 years. George Glenner isolated the amyloid plaque material and figured out what was making those deposits, and that's the amyloid precursor protein and the amyloid peptide. A major, major breakthrough. That breakthrough will lead to the next generation of treatments that we're working on today. So what we have today are some drugs that improve memory by adjusting levels and activity of acetylcholine based on the chemical characterization of the plaques. We are going to move to the next level of treatment, which is not just improving modestly memory, but controlling the pathology of the disease, halting its progression in the brain. Now, even today, the economic burden of Alzheimer's disease is huge. It's estimated at over $100 billion. As the disease doubles and triples over the next few decades, that annual cost will become just astronomically huge. Today, it's the third most expensive disease after cancer and cardiovascular disease. But before long, it will be the most expensive and most important disease. And yet, despite this annual cost, we spend only about $500 million a year on AD research. So that's my complaint for the evening. Here again is the steady increasing prevalence of Alzheimer's disease just on the basis of the aging population. And it means that we have to move beyond the mildly effective symptomatic treatments that we have today and figure out how to prevent this, or it is going to be an unbearable burden on society, not just the suffering of families, but also the economic burden. So this is my message, that this is something that we have to deal with more effectively than we've been able to do so far. And that's what I want to tell you in the remaining quarter of an hour here. Just to give you a little overview of how we're going to move from the modestly effective symptomatic treatments we have today, that is drugs like Aricept and Namenda, to the next generation of treatments which will halt those plaques and tangles in the brain, slow the progression, and eventually prevent the disease. Now, before I move on, I want to tell you a little bit about our group here in the Department of Neurosciences, which is called the Alzheimer's Disease Cooperative Study. The Alzheimer's Disease Cooperative Study, or the ADCS, is the largest program funded by NIH to find better treatments for Alzheimer's disease. It was actually the investigators who conducted the first successful trial of any drug. So the first drug that was tested, which acted on that chemical acetylcholine, was tested not by a drug company, because the companies were not yet convinced that there'd be anything we could do about this disease. It was tested by academic physicians, that is, faculty members at universities with support of the NIH. And that effort was quite successful, and it convinced the NIH to fund on an ongoing basis academic investigators to push the field forward. So even though that first academic trial was successful and it led to many pharmaceutical companies pursuing treatments, and that's how we got Aricept and Namenda, it showed the importance of medical schools and universities in laying the groundwork and making the breakthroughs that could lead to pharmaceutical industry success in bringing drugs to market. So based on that early success, NIH made a commitment to fund a consortium of sites around the country, and that's the ADCS. The ADCS consists of sites around the US. It was started by Leon Thal, who was here at UCSD and was also a chairman of neurosciences. He was recruited here by Robert Katzman, the guy who first put this disease really into everyone's attention. And Leon Thal started the ADCS 20 years ago with the mandate from NIH to move forward the development of therapies for Alzheimer's disease. We have sites around the country and some in Canada. We have 35 main sites and then about 50 or 60 other sites. And we all get together and work on improving our ability to study drugs and actually doing drug trials in Alzheimer's disease. The ADCS is known not just for conducting trials, but for improving the tools that we all need to conduct trials. So back in the 1980s, when the first ideas about treatment were developed, there was no way to test them. Because if you look at someone with Alzheimer's disease, there is nothing that you can see, and there's no way to tell through general observation that someone is getting better or worse over a short period of time. So it took academic investigators with support of NIH to develop the tools. And these tools included memory tests and other cognitive tests and various other ways of assessing the degree of impairment and measuring the impact of therapy. And the ADCS was responsible for developing those tools and then for sharing those tools, not just with other universities around the world, but with the pharmaceutical industry. The ADCS established the methods for doing trials and continues to improve our design of clinical trials in Alzheimer's disease, meaning the ADCS continues to explore new measurement methods, now not just memory tests, but brain imaging tests and blood and cerebrospinal fluid tests to help us study the impact of treatments on Alzheimer's disease. We're now working on a number of trials that bring treatments to earlier stages of disease and explore very novel methods of treatment. For example, gene delivery to the brain. So one of the studies that we're conducting now actually delivers genes for protective proteins deep into the area of brain at which the Alzheimer's degeneration is based. And that involves neurosurgical procedures. So we are exploring all types of interventions to try to bring about the next generation of therapies. The ADCS has conducted over 24 studies over its 20-year history, and some of those studies have been positive, but most of those studies actually have been negative and have had a significant public health impact. So as you know, over the last 20 years, there have been many ideas talked about and influencing behavior of physicians and patients. And the ADCS always takes these ideas and tests them in a rigorous fashion with randomized, controlled, blinded trials. And the results are often very surprising. So for example, I told you that women live longer than men. Women also have a different susceptibility to Alzheimer's disease. And it led to an idea that female hormones, estrogens, might influence the disease, and that estrogen treatment might be useful for Alzheimer's disease. And estrogen was widely prescribed for Alzheimer's disease as a result, but it was the ADCS that conducted the first actual randomized controlled trial of estrogen that showed that it not only was unhelpful, but actually made the disease worse. And as you know, estrogen therapy has had a similar history with cardiovascular disease and cancer. And despite all of the evidence from epidemiology and laboratory studies that estrogen was good for so many things, the actual studies proved that it was more often harmful than helpful. In a similar way, we thought for many years, because there's inflammation in the brain in Alzheimer's disease, that taking anti-inflammatory drugs would be useful. But the ADCS conducted studies of a number of anti-inflammatory drugs, including steroids and nonsteroidals, that showed the opposite, that the trend is to make people worse. And so one of the a major impact of the ADCS on treatment of this disease has actually been negative in the sense of proving that commonly used remedies that carry risk are actually not helpful. Now, our focus today at the ADCS is really on moving to a new type of clinical trial, and I'm going to tell you that story very briefly. So whereas our early studies focused on chemical neurotransmitters and on general treatments like estrogen and anti-inflammatory drugs, our efforts today are based on the developing science, understanding why there are plaques and tangles in the brain. So in this cartoon, you see these plaques and tangles, and we now understand the biochemistry of plaques and tangles. So this is a drawing of a protein, a big macromolecule that sticks through a cell membrane. And we now know that this fragment of this normal protein is the toxin that forms these plaques and also damages nerve cell function, damages structures within the cell, and eventually leads to the formation of tangles. The science of Alzheimer's disease at the molecular and cellular level is actually summarized in this cartoon, and this cartoon is the basis of our new efforts to treat this disease. So we now believe with confidence that the actual cause of the disease, so aging is a big risk factor, but the actual molecular cause, what goes wrong chemically in the brain that leads to damage of cells and plaque deposition and tangle formation and dementia, starts with a specific protein called APP. That's this protein. So this is a drawing of a protein that sticks through a cell, and within that protein is this fragment. Here's the protein called APP. Here's the fragment called A beta. And we believe that getting that fragment out of this protein is what sets the disease on its way. That this is the inciting event in Alzheimer's disease. And then everything, including the death of brain cells, follows from this step right here. And the reason that we are very confident that this is correct, even though there are many, many abnormalities in the brain, we're confident that this is what starts the process. This is the key to the disease, because there are a number of genetic forms of Alzheimer's disease. There are genes that can cause a very early form of the disease, and every gene that's been associated with Alzheimer's disease has now been shown to directly influence this step. So if you are in a family that has a genetic mutation to cause Alzheimer's disease, you have a mutation that influences the cutting of this protein to release this fragment. And there's really only one way to interpret that. It means that this is what's causing the disease in those families, and we believe this causes the disease in everyone. You may know that people with Down syndrome have a very high risk for Alzheimer's disease. In fact, the risk approaches 100% by middle age in people with Down syndrome. Now, why is that? Well, Down syndrome is trisomy 21, an extra copy of chromosome 21, and it turns out that the gene for this protein is on chromosome 21. So people with Down syndrome just have 50% too much of this gene, and that pushes through this pathway to give you amyloid plaques and neuron death and dementia. What that means is we know what we need to do. So once you have a specific cause, you can design specific therapies. Now that may sound easy. It's not easy at all, but it is possible. And when you're dealing with a problem as enormous as Alzheimer's disease, you need to put your resources into translating this understanding of the molecular basis of the disease to effective treatments. That's what we're trying to do now, and I'll tell you a little bit more about it. But the more funding we get, the more investment in this, the faster we get there. So we have great targets. We just have to get there. We have to translate those targets into better therapies. We need to find specific treatments to inhibit these enzymes that release this toxic fragment from the parent protein, or we need to find ways of simply clearing that fragment from the brain. And these enzymes that release the toxic fragment can be inhibited, and we have to find safe, effective inhibitors that can get into the brain, or we have to attack this fragment, and it turns out that the best way to attack that fragment is actually with antibodies and vaccines. Now, there have been a number of breakthroughs just in the last 5 years that make us confident that we're on the road to developing better treatments that get at that inciting pathway. I've just shown one here. So this is a new form of brain imaging called amyloid PET imaging. It's not like MRI scans. It's a way of not looking at the overall structure of the brain, but rather looking at the accumulation of amyloid plaques in the brain. Up until this test, which is currently being reviewed by the FDA and will probably be approved for clinical use sometime in the next few months, up until this test was developed, There was no way to tell that someone had incipient Alzheimer's disease and no way to track the progression of the disease. Now we have a test that shows amyloid in brain very accurately. This allows very, very early diagnosis and allows us to assess the impact of treatment. And this tool itself is a big step forward in the development of treatments to actually block this amyloid deposition. Now, we've moved forward with a number of treatments aiming to reduce amyloid, and they've all failed so far, and that's the bad news. Now, they failed in part because we didn't have the tools to design the trials effectively. We didn't have the brain imaging to make sure that we were doing what we thought with anti-amyloid treatments for people with Alzheimer's. But now that the tools are better, we think we're going to move beyond these failed trials and increase the likelihood of positive trials. The effort is substantial. I don't want to make it sound easy. We have a lot of confidence that we have a good target, but it's been so difficult because you need to inhibit these enzymes or attack this amyloid in a very specific way. And you're talking about changing brain function, and that's quite hazardous in an older person. So developing safe and effective therapies is challenging. We're constantly searching for better drugs, but we actually think that our refinement of methods and our ability to move treatments to an earlier stage of disease is going to markedly increase the likelihood of our success. Now, what do I mean by moving to an earlier stage of disease? Today, when you diagnose Alzheimer's disease, you do it on the basis of the syndrome of dementia, where there's cognitive impairment significant enough to change daily function. What we now know through using the tool I just described, that amyloid PET scan that shows amyloid in the brain, we now know that amyloid accumulates in the brain before the dementia syndrome starts. How much before? 15 years before. There is a 15-year gap between the onset of the pathology in the brain and our ability to diagnose it using just cognitive tests and symptoms. Now that we have amyloid imaging, as well as tests where we look at cerebrospinal fluid, we can identify people here, and we don't have to wait until here to initiate treatment. So what that means is that the point of diagnosis today is really an end stage of disease. Now, if you think about a degenerative disease of brain, something that's eating away at the structure of brain, if you want to have a big impact, you want to initiate treatment before you've lost much. And being able to identify people 15 years earlier is a huge step forward. So in the community of Alzheimer's researchers, we are moving away from the standard diagnosis today to describing and identifying people at very early stages, much closer to the onset of the brain pathology, to the onset of the plaque accumulation. And this is, again, a 15-year march earlier. And now we're actually working on identifying people at a pre-symptomatic stage with no symptoms on the basis of abnormalities that we can measure in spinal fluid and image in the brain. I'll just mention that this study of biomarkers, of markers that allow us to identify people before there are symptoms, has been hugely moved forward by an initiative called ADNI, Alzheimer's Disease Neuroimaging Initiative. I mention this also because it was also started by Leon Thal here at UCSD. The principal investigator is now Mike Weiner at UCSF. It's funded by NIH. This is everybody getting together to try to understand these imaging methods and biochemical methods of early diagnosis. And it's an amazing development in science. This slide shows you the groups that are contributing to this study of biomarkers in Alzheimer's disease. It is a difficult problem, it's an expensive one, but we've been hugely successful in demonstrating how biomarkers can lead to early diagnosis and early tests of therapy. All of these companies are contributing to this ADNI study and openly sharing all data. And this level of collaboration, you know, you read a lot about companies being only interested in profits, and I'm not going to, obviously companies are interested in profits, but for a disease like this, they also recognize that for us to make progress, we have to work together. And the degree of collaboration in this field is just amazing. So ADNI has been supported by all these companies, plus NIH, plus philanthropy. And it has led to amazing developments, including amyloid imaging, over the last few years. This is a model that we've put together using data from this ADNI collaboration that basically tracks the trajectory of many biomarkers that allow us to have a signature of Alzheimer's disease even at the asymptomatic phase, to use spinal fluid and brain imaging to identify people years before they have symptoms. And this is where we're now focusing our therapeutic trials, our clinical trials. This is just an example of using imaging, amyloid imaging and MRI imaging, to track the progression of disease in people who have no symptoms and to demonstrate that amyloid is causing the problem. And this type of experiment, which comes from ADNI, gives us the specific framework for testing drugs in asymptomatic people who are headed for Alzheimer's disease. And so we're just getting started on this type of study where we screen not people who have memory problems, but cognitively normal older individuals with amyloid PET imaging or spinal taps, and then begin treatment years before symptoms and use imaging to track the effective treatment. And this is going to allow us to take our anti-amyloid therapies to the stage of illness at which they are likely to work. What will this mean for the future? So now this is purely projection on my part. But what I think will happen is, now that we understand amyloid, we understand that it's causing Alzheimer's disease, and we can measure it in people with no symptoms, we will end up screening older individuals, and we now know that amyloid deposition in people who are going to develop AD starts around age 55. So just as doctors recommend colonoscopy now to screen for colon cancer, we're going to be screening for amyloid. And when amyloid becomes positive, we're going to treat the amyloid accumulation before there can be damage to brain cells and brain function. And this is the long-term view of how we end up preventing Alzheimer's disease, based on this scientific collaboration giving us the tools to take our anti-amyloid interventions to the earliest possible stage of disease. How long it will take us to get here from where we are today depends just on how many resources we put on this problem. We will get here, we'll get to this point maybe in 20 years, but if we put more resources into it, we could get there in 5 years. So that's our hope. And with that, I'll just acknowledge all of the people who work on these studies and our funding agencies and investigators, especially the thousands of families that volunteer to participate in our clinical trials, because without clinical trials, we'll never get anywhere. So with that, I thank you for your attention.